Welcome to this biology lesson in photosynthesis. In this lesson, we're going to be mainly looking at the factors that affect the rate of photosynthesis. When we say rate of photosynthesis, we mean how fast photosynthesis happens in green plants and algae. As you know from earlier lessons, algae is a single-celled organism which that has got a lot of common characteristic features, design features with plants. So they can do photosynthesis just like plants can. Okay, so they should have in their cell structure something that is known as chloroplasts. We know this from our earlier lessons that chloroplast contains chlorophyll. So I'm going to write a chloroplast which contain chlorophyll. And chlorophyll is the chemical substance that is present in the green pigment chloroplast. In fact, green plants and green algae, they look green because of this green pigment chloroplast. What do? The main function of chloroplast is to trap sunlight energy so they can do this chemical reaction known as photosynthesis. So this lesson is about an introduction into photosynthesis. What is this chemical reaction, which is so very important to all living things in more than one way. And secondly, we're going to be looking at something called limiting factors. Before we go into limiting factors, we need to know what are the factors which affect the speed of photosynthesis, and then we go into limiting factors. So what is this photosynthesis? It is basically a chemical reaction, and it's such an important chemical reaction because it helps all other living things, anything other than plants, in providing food. Okay, so what does it involve? There are two main substances, carbon dioxide and water. These are the two substances that the plants use and to produce glucose. Glucose is the food it makes, and the organ that this mainly happens is the green leaves. But it also happens in other parts of the plant, such as the stem, which are green. If it is green, it also happens there. Now, most of the plants don't have green stems. So main organ where this reaction happens is the leaves. Okay, and the leaf is specially adapted for that purpose. We'll see that in another lesson. So for now, we need to know the reactants in these chemical reactions are carbon dioxide and water, and it produces glucose and oxygen. We're going to see much more about glucose later on, but for now we need to understand that this is the basic chemical that makes starch, which is found in many starchy food, potatoes, and also it is used to make other substances such as protein, oils, fats, cellulose. So for everything else that we have as food and the plants make themselves, this is the basic raw material or the starting material to build all those things. So we're going to be learning about how those substances are made by the plant itself. Sometimes the plants are eaten by animals and then they make different types of protein. So we're going to be looking at how plants convert glucose into other substances later on. Another useful substance, the most essential substance for animals, are also made by plants in this process, which is oxygen. Okay, so as we know, without oxygen, animals cannot survive. Okay, it is important to realize at this stage that the plants actually make glucose, which is a store of energy, by converting or by using sunlight energy. So plants cannot perform this photosynthesis equation without that energy from sunlight. So it is important to realize the energy comes from the sunlight, which gets stored in the glucose, which is essential for other animals to survive and for the plant itself for its energy source. Glucose is sometimes also referred to as sugar. So Sugar is just a bigger molecule made from glucose, and sometimes it is referred to in textbooks as sugar is made by plants. So you need to understand that it is actually talking about glucose because it is only a smaller unit of sugar. So why is it called photosynthesis? Let's have a look. Photo means light. Synthesis is making normally a substance. So because the plant makes glucose using the light, it's termed as photosynthesis. So that's the connection. Let's now have a look at how this is connected with another very important chemical reaction that all living things derive their energy from. You must have heard about respiration. All living things, including bacteria, most type of bacteria, and many other living things, algae, plants, including animals, we all do perform this chemical reaction at a cellular level in the cells, in all the cells of our body. This reaction happens. This is the one which provides us with the energy, the energy to do our lives function. So that all comes from glucose. Now, if you look at this glucose, that is actually made by the plant. So what we do, if you have a look at this equation, it is just the reverse of the respiration reaction is just the reverse of that reaction which you see here. If you just swap the reactant for products, you get this. Glucose and oxygen is over here. So all living things make use of glucose and oxygen from the air to produce the energy. As you can see, energy is needed for photosynthesis, but energy is produced by respiration process. But the substances involved are just the same, but the equation is swapped and it's reversed. I think at this stage it's worth knowing how the plant gets the basic substances involved in photosynthesis. Water is absorbed through the roots and it's transported through the stems to the leaves. And carbon dioxide comes from the air and it's taken in by diffusion. Carbon dioxide just moves into the leaves through the holes at the bottom of the leaves, which are called stomata. It moves in through these tiny pores into the leaves, and the leaves has chloroplast, which contains the chemical chlorophyll. So leaves contain chloroplast. This contains the chemical chlorophyll, which can actually trap the sunlight. So the main function of chlorophyll is to trap the sunlight energy to carry out the photosynthesis chemical reaction. To understand limiting factors, it's useful to think of an analogy, which in this case I want to consider making a very simple cake. Don't think of anything complex. It's a very simple lemon flavor cake. And to make this, you need these ingredients: 4 eggs, 1 pack of flour, and half a pack of sugar, half a pack of butter, 1 teaspoon of flavor. So in this case it's lemon flavor. Okay, we have now these ingredients and this is how much we've got. Okay, so we've got like 12 eggs here. 12 eggs. We have unlimited supply of flour. Okay, so that's there. We have more than enough flavor, lemon flavor. And we have 3 packs of butter. And we have 5 packs of sugar. Okay, so if you start making the cake, you can see from our recipe we need 4 eggs per cake, right? So I'm going to cross out as I keep making the cake. So 4 eggs gone. That's the first cake. And 1 pack of flour, half of sugar. So we're going to put half exactly and scrub it out. Okay, with half is gone and half of the butter is gone. Okay, I've only used very little amount of flavor, so there's plenty left now. Okay, so we don't have to worry about the flavor for the moment because we got lots left. Okay, so second cake. So I've made one cake. Now second cake. Second cake, another set of 4 gone. The other half of the butter will be gone, so the whole of that is gone. I hope you're following this. Another whole pack of flour gone, and now we've got used 1 whole pack of sugar. Okay, now third cake, another 3 eggs. Another 4 eggs. Another half of one pack of this flour, half of this butter. So I'm removing half of that. Is gone. I hope you're following this. We still have 1.5 pack of butter left. We got plenty of flavor left. Okay, so we are on to the third cake. So we also need to scrub out half of this sugar, half pack of sugar we need for each cake. So we've made 3 cakes, as you can see. 1, 2, 3 sets of 4 eggs gone. We've used 3 packs of this. We have plenty of flavor left. And so, can we make any more cakes? So the maximum we can make is 3 cakes. I hope you understand this. In this case, what we run out of first, which is limiting the number of cakes, is not the butter, because we got loads more butter left. As you can see here, we got like at least enough for another 3 cakes. We got unlimited supply of flour. We got lots of flavor. We got some lots of sugar more, but the limiting factor in this case is the eggs. The eggs is the one factor which is now stopping any more production of the cake. So you can't make any more cake because you're limited with the supply of eggs, and that's why we call that as limiting factor. So now we go back to our photosynthesis, try to understand what limiting factor is. It'll make hopefully slightly better sense, because now we know what a limiting factor is. As in this case of the cake, the limiting factor, as we know, are the number of eggs that we have. Simple experiments as shown here on this diagram can be done in a lab, in a school lab, to show how different factors affect the rate of photosynthesis. As you can see, you have a desk lamp here. You have a plant which is known as Elodia. It is actually a weed which grows in ponds. It's pond weed, commonly known as. And you have thermometer to measure the temperature. You can adjust the temperature of the water here. That will affect the temperature at which the photosynthesis takes place. And this is one of the factors. You have carbon dioxide, which is a chemical that you can add on, such as bicarbonate. You can add on to water to produce carbon dioxide, or to provide carbon dioxide for photosynthesis. You can adjust the intensity of the light, so you can adjust the light intensity in that way. So by doing this experiment or similar experiments, what you can do is you can affect the main factors that affect the rate of photosynthesis. So these are the factors which we are going to be focusing on, which is light intensity, the carbon dioxide availability, how much carbon dioxide is available for the plants, and the temperature at which the plant is carrying out the photosynthesis. Sometimes it's very cold in the winter season, so and in summer it's rather warm. So how this factor affects the rate of photosynthesis is something that we can investigate and find out, and plot the results in the form of a graph. Okay, so let's go back to the experimental setup. Here, as you can see, the temperature is at 40, it's set at 40. A certain amount of carbon dioxide is added to the water in the form of a chemical, normally a carbonate, which easily decomposes in water to provide the carbon dioxide for the plant. Light intensity is set at 5, and see, compare this one where you have same temperature, same amount of carbon dioxide, but the light intensity has been increased to 10. With this, now light intensity has increased to 40, has been increased to 40. Obviously, you can systematically increase this to from 10, 20, 30, 40, or if you don't have a control to control the intensity of the light, you can just adjust the distance of the desk lamp to control the intensity. So if you move it further, you have lower intensity light, and if you move the lamp closer to the Lodia plant, you have a higher intensity light. So you can actually control just the light intensity with keeping the other factors the same. In other words, these are the control variables. So when you change only one thing, which is your independent variable, you keep the other two factors the same to carry out experiments such as these. So in this case, the independent variable is the intensity of light. The control variables are the temperature at which the photosynthesis takes place and the level of carbon dioxide, or the amount of carbon dioxide, or in other words, the concentration of carbon dioxide in water. Okay, so what is the dependent variable? Dependent variable obviously is the rate of photosynthesis, but how do we actually measure it? Is by measuring the number of bubbles the Elodia plant makes. And so as we know, plants during photosynthesis make oxygen gas, and which comes as tiny bubbles through the water. So by just counting the number of bubbles per minute, we should be able to find out the rate of photosynthesis. So following on from that experiment that we've just described, we have the rate of photosynthesis measured by the number of bubbles that was produced in that experiment. So independent variable is the light intensity, dependent variable is the rate of photosynthesis measured by the number of bubbles that has been made. So let's make sense of what are limiting factors in this case. So as you start from here, the light intensity increases, the rate of photosynthesis increases up to a certain point. Now, as you can imagine, from this point onwards, the rate of photosynthesis does not increase anymore. It stays level. It levels off, and as so you can see that there is something that is affecting the rate of photosynthesis. It doesn't increase anymore, but there is plenty of light available. You keep on increasing the light intensity, so there is no shortage of light. So the only limiting factors in this case, it could be the level of carbon dioxide or the amount of carbon dioxide that's available. So amount of carbon dioxide, or temperature. So you can say that it could be one of these factors which is a limiting factor past this point. Okay, so if you want to increase the level of photosynthesis any further, you need to increase one of this amount of carbon dioxide or available to the plant, or you can increase the temperature to see if it increases the rate of photosynthesis any further. So just to summarize that, the limiting factor when at this point in the graph is, it could be one of these two or both of them, amount of carbon dioxide and temperature. As you have unlimited amount of light intensity available still, but it's not helping the plant to increase its rate of photosynthesis. But here at this point, the light intensity is the amount of light, or the light intensity is a limiting factor. As you can see, as you increase the light here, the rate of photosynthesis increases. So here it is limiting, but when it comes here, light is no longer a limiting factor. So it is one of these two. The reason is plenty of light is available past this point, because as you're still increasing the light intensity. But you can see it's one of these factors which affects the rate of photosynthesis to go up any further or any higher. So what if you keep the light intensity and the temperature at a constant rate? You keep them controlled, but now you are increasing the level of percentage of carbon dioxide available to the plant. Again, you can see a similar shape of the curve. It increases as you increase the level of carbon dioxide initially up to this point. Up to this point, it responds to the increase of carbon dioxide. So that means over here, carbon dioxide is a limiting factor, because as you increase the carbon dioxide, it's still responding by increasing the rate of photosynthesis. Okay, but at this point, so what I mean is over past that point, the increase of carbon dioxide has no effect on the rate of photosynthesis. So what that means is, past this point, carbon dioxide is not a limiting factor. That's not stopping it going up any further. It's actually the temperature or the light intensity. So past this point, you can increase the light intensity or the temperature to make the graph go up slightly further. Okay, so if you increase one of these factors, like say temperature, so if you increase the temperature past this point, the rate of photosynthesis may further increase and then level off. Maybe at this point, the other one becomes the limiting factor, which is the light. So by increasing each factor, you can increase the rate of photosynthesis slightly. Let's have a look at how increasing the temperature affects the rate of photosynthesis. So just to summarize this graph here, you're looking at percentage of carbon dioxide increasing on the x-axis and the rate of photosynthesis increasing initially, which means over here carbon dioxide is a limiting factor. Past this point, as further increase of carbon dioxide has no effect on the rate of photosynthesis, you can say carbon dioxide is no longer a limiting factor. So past this point, it's actually the light level, the light intensity, and the temperature are limiting, which means they are stopping the photosynthesis from going any higher. So past this point, say if I increase the temperature slightly, the rate of photosynthesis may increase slightly, like shown here, but then it levels off because it reaches the maximum again for the light intensity available. So at this point, further increase of temperature may not have an effect. You need to further increase the light to have an effect, to increase the rate of photosynthesis a little bit further. So I have written this in here now. Carbon dioxide is the limiting factor at the start because it is still increasing when you increase the carbon dioxide. Past this point, as you can see, carbon dioxide is not limiting, but light and the temperature are limiting factor in this part of the graph. This graph shows the effect of increasing temperature on the rate of photosynthesis. So as you can see here, at the start temperature is limiting because as you increase the temperature in this area up to 40, the rate of photosynthesis does increase. So here temperature is limiting. Remember, you have controlled the level of light available or the light intensity. You have also controlled the concentration of carbon dioxide available to the plant or the amount of carbon dioxide. So up to this point temperature is limiting, but after this something strange happens. It suddenly falls down, it decreases. So up to 40 degree of temperature, the rate of photosynthesis does keep on increasing, but after 40 it suddenly reduces to nearly zero. That's because you have in the plants a protein called enzymes, which are really essential to carry out the photosynthesis. And what happens is these enzymes denature at higher temperature. What that means is they lose their shape, and because of this it is not able to photosynthesize effectively after 40 degree temperature. So plants are affected of beyond 40 degree. If the temperature goes above 40 degree, it's not able to photosynthesize. You can also see from the graph that the optimum temperature for maximum photosynthesis is 40 degree C. As you can go down and see, that's the best temperature to use to maximize crop production, particularly inside greenhouses, which is what we're going to be looking at next. So these ideas of limiting factors are actually used in the commercial production of food in the greenhouses. So we're going to be looking at how it is really industrially important, and for limiting a study of limiting factors and an understanding of what affects photosynthesis is really important for us in terms of food production. So as we know so far, light intensity can affect the rate of photosynthesis. Amount of carbon dioxide is a factor and temperature is a factor. So what people do, particularly those who produce food in greenhouses, they adjust these values, light intensity and the temperature and the amount of carbon dioxide, to an optimum level to maximum use by plants for photosynthesis, and in turn to produce maximum amount of food. So if these conditions are optimal, then food production is maximum. So let's see how that is done in practice. Most of the food nowadays is being produced inside greenhouses, like so. You have a large enclosure of glass houses in which you can have crops grown intensively under controlled conditions of light and the temperature and the level of carbon dioxide. All can be adjusted in here, or adjusted by the producers, the farmers, to maximize their profits in a way, and also to maximize the food production. So as you can imagine, these three factors are really controlled inside the greenhouse. Let's see how that is. So that's the greenhouse, and what you see here is a heater, and you can pretty much guess why this heater is there. That is to produce the optimum temperature and the warmth of about 40 degrees C. As we have seen from the experiments before, that gives the best yield of crops. So that heater gives the optimum temperature. The lights can be adjusted, the light intensity, depending on how much sunlight is already available, and also the carbon dioxide level can be adjusted inside to an optimum level. It is constantly measured by computers inside. Sensors measure the temperature and the carbon dioxide levels continuously, and sometimes in some cases smaller greenhouses actually use fuels, hydrocarbon fuels. As you know, by burning hydrocarbon fuels you make carbon dioxide. So by just using a fuel heater instead of electrical heaters, you do two things in one. You produce the carbon dioxide necessary for the photosynthesis, or a correct level of carbon dioxide, or you increase and maximize the level of carbon dioxide, and also produce the temperature required by just using a heater which uses hydrocarbon fuel. I can now show you a very simple version of a heater which uses hydrocarbon fuels. So there you can see a small stove-like equipment which burns the fuel over here, and because as this is a hydrocarbon fuel such as paraffin, it produces carbon dioxide. So the level of carbon dioxide here is really high. Again, this can be controlled. After a while, plants only can use a certain amount of carbon dioxide, and also people have to work in here, so you can't have too high level of carbon dioxide in here. so this can be switched off and on as required. Or in modern greenhouses, this is all controlled by computers to maximize the food production as well as the safety of people who work there. So just to summarize how the limiting factors are actually put to use commercially in the case of greenhouse is this. They control the light intensity to maximize the rate of photosynthesis, light intensity and the level of carbon dioxide or the concentration of carbon dioxide in the air, and the temperature. Temperature, as you can imagine, should be kept at 40 degrees C, as we know that is the best temperature for photosynthesis. That is the end of this lesson, and in the next lesson we can look at how plants use the product of photosynthesis. For instance, it produces glucose. What happens to that glucose? How the plant stores it in different forms. That's for next lesson.